more than 1 billion of bacteria just in 1 gram, let's say, or a small amount of soil. So with these methods, we only can isolate or can describe the 1% of the real biodiversity, since most of this biodiversity is not possible to culture, no? Under lab condition. So this is one of the limitations of this method. But on the other side, with these methods, we can also isolate some microorganisms that can be potentially used for another biotechnological practice, like they are producing some enzymes, or they can be some plant promoting growth, and they can be used in plants. Another direct method should be can be like enumeration using fluorescence microscopy. In this method, we can count the bacteria because it has been previously dyed using different dyes like propidium iodide or DAPI, and it allows to determine the quantity, or the enumerated again by direct methods, the quantity of bacteria. It's a very simple method because you need to use one dye, and by computerized image analysis you can see what do you have in your sample. If we move on to the indirect methods, we have to measure the microbial biomass. I include here also the DNA measurement based on, or the qPCR based on DNA measurement, because we can also have an enumeration of the bacteria in soil according to the number of genes of bacterial or fungal communities. If we isolate previously the DNA and we just try to amplify the copies of these genes by this technique. In these indirect methods, I'm going to probably mention some that already have been mentioned by my colleagues, but I will be very fast. So also for microbial biomass, we can do by chloroform fumigation methods. That this is a physiological methods for quantification of microbial biomass, one more time, and it's based on the quantify of the vapors once that the soil is fumigated with chloroform. So this chloroform will kill all the microbes in the soil, and it can be quantified the dead biomass, quantifying the amount of carbon dioxide. Another method is the substrate induces respiration. That this method is based on the principle that is at the increasing level of glucose, it's going to increase the rate of respiration in soil, and it's going to be directly proportional. This method can measure the change in the initial soil respiration rate after addition of glucose, because you need to add the glucose to measure this respiration. And it's a method also can allow you to distinguish between the fungal and bacterial component if you are using different inhibitors. Another method to quantify the biomass is the microbial ATP content. In this case, the microbial cell can be disrupted rapidly with the help of suitable reagent. So the ATP is released and stabilized by the addition of some enzymes, and is extracted from the soil matrix. And the limitation of this method is that ATP can be easily degraded, so sometimes not very reliable method to this determination of biomass. To measure the microbial biomass, we can also do it by freeze drying the soil extraction. So it consists like you freeze dry the soil, and after that you can add some sodium carbonate or sulfate potassium, and it's going to release the carbon compounds and the disrupted microbial cells that are going to be extracted and colorimetrically measured. This is rapid colorimetric methods, and it's safe and precise. In the last note, this is almost the last one, the UV spectroscopy method, that is consists in the measure spectroscopy measure of the DNA of nucleic acid in the sample of soil. And and it can have some limitation because the soil can produce some interference when you are measuring by spectroscopy because some colloidal part or suspending particles. And in the last of these, I have two more. There is another method that is rehydration methods. It consists the air-dry soils are rehydrated with water and dilute in salt solution, and it's allowed to determine the microbial biomass because due to the rehydration, the desiccated microbial cells are disrupted and they can be measured by potassium dichromate oxidation methods. It will give information about the biomass content. And in this section about microbial biomass parameters, I include also ergosterol, that is a compound that is in the membrane of fungi. So it can be extracted by a procedure with some solvents, organic solvents, ultrasound bath. And it can be quantified by HPLC, and it can give us a parameter about the fungal biomass in soil of this, according to this method. It can be useful for fungal markers or also to quantify mycorrhizal, arbuscular mycorrhizal. And with this method, I'm finishing the part of microbial biomass. So the next part is the method according to microbial activity. And in this part, we are going to focus in the method